After studying this module, you shall be able to learn about the natural process of dinitrogen fixation, which is the biological nitrogen cycle, the role of nitrogenase system in dinitrogen reduction, the structure of various dinitrogenase systems and the locations of various metal complexes in it, the role of metal complexes in dinitrogen reduction, and also you will come to know about the proposed nitrogen cycle. Most of the chemical species involved in biological systems, such as including proteins, nucleic acids, chlorophyll, various enzymes and vitamins, and many other cellular constituents contain nitrogen. Hence, a source of utilizable nitrogen is an essential requirement for all the living things, which generally comes in the form of ammonia, NH3, and nitrate, NO3-. The Earth's atmosphere is the principal reservoir of nitrogen, where it is present in the form of dinitrogen as the most abundant constituent of the atmosphere. Although the most abundant constituent of the atmosphere is dinitrogen, the unreactivity of dinitrogen limits its usefulness. The nitrogen requirements of the biosphere are thus provided by a process called nitrogen fixation, which involves biological reduction of dinitrogen to ammonia. A small amount of dinitrogen and dioxygen reacts abiologically during lightning discharges and high temperature combustions. The biological reduction of nitrogen is carried out exclusively by prokaryotes, including various bacteria such as blue-green algae, yeasts, and in symbiotic bacteria legume associations under mild conditions. The molecular nitrogen is so highly unreactive to normal chemical reactions that it is considered as almost as inert as a noble gas. Here we can see the structure of dinitrogen, which contains a triple bond between two nitrogens, whereas the energy of this triple bond is around 945 kJ per mole. Dinitrogen is isoelectronic with carbon monoxide. Carbon monoxide has a filled lone pair orbital on the carbon atom of sigma type. Therefore, it can form sigma bond with metal ions. Carbonyl also has empty pi star orbitals, and hence it has the ability to form back bonding with electron density from metal to carbonyl. On the other hand, dinitrogen also has filled lone pair orbitals of sigma type, but they are of very low energy than those of the corresponding orbitals of carbonyls. Because nitrogen is more electronegative than carbon, hence dinitrogen is a weaker sigma donor as compared to the carbon of carbonyl ligand. The empty pi star orbital of dinitrogen are lower in energy than those of the carbonyl group. Hence, dinitrogen display bank bonding properties more readily than carbon monoxide. As a result of these bonding properties, dinitrogen binds less efficiently than carbon monoxide with metal ions in its metal complexes. Here we can see the structure of carbon monoxide, which is also containing a carbon-oxygen triple bond, and a structure of dinitrogen, which contains a nitrogen-nitrogen triple bond. Here we are summarizing the bonding facts. Now, in vivo dinitrogen fixation. The first example of dinitrogen complex, which is the pentamino dinitrogen ruthenium dianion, where the anion is either bromide or iodide or hexafluorophosphate or tetrafluorophosphate, was separated from the reaction of ruthenium trichloride with hydrazine in the aqueous solution in the presence of different anions. This was synthesized by Allen and Zenoff in 1965 while other complexes such as the cobalt analog was prepared directly from gaseous dinitrogen. Here we can see some of the examples. The ruthenium example is the first example of transition metal dinitrogen complex, whereas the cobalt one is the first example of transition metal dinitrogen complex which was derived directly from molecular dinitrogen. The molybdenum complex is the first example of the molybdenum dinitrogen complex. While the synthesis of these initial metal complexes, it was discovered that molecular dinitrogen is capable of forming stable metal complexes with transition metal ion. Since then, several attempts have been made to study the behavior of molecular dinitrogen as a ligand towards various transition metal ions. One of the most successful industrial processes of dinitrogen fixation is called as Haber's process, which includes catalytic reduction of dinitrogen over an iron aluminum trioxide catalyst at a high temperature of around 500 degrees Celsius and high pressure of about 300 bar. Here we can see the reaction which is involved in Haber's process, which includes the combination of one molecule of dinitrogen and 3 molecules of dihydrogen under the specified conditions to form 2 molecules of ammonia. In the another method, the titanium alkoxide complexes were found to react with dinitrogen to form stable complexes which subsequently release ammonia or hydrazine on reduction. The reaction can be shown here. Certain molybdenum and tungsten complexes also displayed the conversion of dinitrogen to ammonia in acidic solutions. These complexes contain phosphine ligands and are of general formula bis dinitrogen and tetraphosphine metal, where metal is generally molybdenum and tungsten, and the alkyl group and aryl groups are there on the phosphine ligand. We can see that this complex undergo this reaction in the presence of sulfuric acid to form ammonia and dinitrogen and several other products. So these type of complexes disintegrates into various products. Hence the dinitrogen is bound to the metal ion and the dinitrogen reduction takes place by mechanism. The dinitrogen first binds with the metal ion and in the presence of an acid it is reduced to the ammonia, followed by simultaneous reduction of the second ammonia molecule. Now we will see in vivo fixation. The bacteria and blue-green algae that can fix nitrogen in vivo are Clostridium pasteurinatum, Klebsiella pneumonia and Azotobacter vanillandi. The most important nitrogen fixing species that can catalyze nitrogen fixation biologically are Rhizobium, which is found in root nodules of various species of legumes such as clover, alfalfa, beans, peas, etc. These carry out nitrogen fixation under essentially anaerobic conditions. Here we can see a proposed biological nitrogen cycle where the nitrate ion is reduced by nitrate reductase, which is a molybdenum complex, to form nitric oxide. Further reduction of nitric oxide by nitric oxide reductase forms nitrous oxide. The nitrous oxide is then further reduced to dinitrogen by nitrous oxide reductase, which is by various other processes is converted to ammonia and its other analogues. So we call this as biological nitrogen cycle. Now we will see what is a nitrogenase system. The nitrogen fixing bacteria contain an enzyme called nitrogenase which catalyzes the reduction of dinitrogen to ammonia. The reaction is coupled with hydrolysis of 16 ATP molecules and production of dinitrogen. Here we can see the reaction involved. The dinitrogen reacts in the presence of 8 H plus ions and 8 electrons to give 2 molecules of ammonia with simultaneous production of 1 molecule of dihydrogen. Nitrogenase has attracted much attention from the scientific community due to the fact that it can reduce nitrogen under normal conditions despite the unreactivity of dinitrogen. On the other hand, industrial production of ammonia is done at the conditions of high temperature and pressure, as we have seen in the Haber's process. The production of this ammonia is extremely important for the biosynthesis of amino acids. The other nitrogen molecules that come out of the nitrogen cycle are nitrous oxide, nitric oxide, nitrate, etc., where nitrous oxide NO serves as a crucial cell signaling agent and a potential greenhouse gas. Till date, a total of 4 nitrogenase systems have been discovered in bacteria and archaea. Most of these systems are based on either molybdenum or iron, whereas few of these systems have been identified to be based either on vanadium, iron or iron alone. Now we'll see the structure of nitrogenase. Nitrogenase is a complex enzyme consisting of 2 types of proteins. The large protein is called the MOFE protein, whereas the smaller one is called the Fe protein. The Fe protein contains a 4 iron 4 sulfur cluster which is coordinated with 2 cysteine residues and has a molecular weight of 57,000 to 73,000 Dalton. The iron protein transfers electron to the molybdenum iron protein by a known mechanism via ferredoxins to the nitrogenase system, where each electron transfer is accompanied by the hydrolysis of 2 ATP bound molecules bound to the iron protein. The structure of iron molybdenum cofactor is most easily described as a 4 iron 3 sulfur partial cubane structure which is bridged to a molybdenum 3 iron 3 sulfur partial cubane through 3 iron bridging sulfide atoms. The molybdenum iron protein consists of 2 types of superclusters, where the P cluster is thought to be an electron transfer center and the other cluster is thought to be the site at which the reduction of dinitrogen to ammonia takes place. The molybdenum iron protein has a molecular weight of 2,20,000 to 2,40,000 Dalton, containing 2 molybdenum atoms, about 30 iron atoms and about 30 labile sulfide ions. It can be speculated that the presence of 6 trigonal iron atoms in the middle of the iron molybdenum cofactor acts as a potential binding site for the substrate, which is dinitrogen. The binding of other substrates cannot be accommodated within this core, so it is highly specific towards dinitrogen. However, the structure of the iron molybdenum cofactor can be evaluated and rationalization of the mode of dinitrogen binding or its mechanism of stepwise reduction at this site is still unknown and requires a lot of effort. Now here you can see the structure of nitrogenase showing the iron protein and the iron molybdenum protein, where the arrow indicates the dinitrogen binding site. Now this is a structure of P cluster. However, the mechanism of dinitrogen reduction is still unknown. It seems likely that the dinitrogen binding involves molybdenum atoms and the overall catalytic cycle may be resembled as shown in this figure here. The dinitrogen is assumed to bound to the molybdenum atom, where it undergoes various reduction cycles which finally ends up with the liberation of ammonia. The reduction process is accompanied by the pyruvate electron pool, which provides electron at every step of conversion of dinitrogen to ammonia. It also provides the H plus ions. So you can see the proposed nitrogen cycle. The leg hemoglobin is a monomer oxygen binding molecule which closely resembles myoglobin. It also binds any oxygen tightly and thus protects the nitrogenase in order to maintain anaerobic condition. This is because nitrogenase cannot operate in the presence of oxygen. Now we can summarize our chapter. The molybdenum iron nitrogenase is the metalloenzyme responsible for the fixation of the atmospheric dinitrogen by reducing dinitrogen to ammonia. The reaction which takes place is coupled with hydrolysis of 16 ATP molecules and also results in the production of one molecule of dihydrogen. The molybdenum enzyme consists of two components. First is the iron protein, which has a molecular weight of 57,000 Daltons and contains iron and sulfur, whereas the second component is the iron molybdenum protein, which has a molecular weight of around 2,20,000 Daltons. It contains both the metals, one atom of molybdenum and 32 iron atoms. The molybdenum iron protein binds the dinitrogen and the iron protein accepts electron from the external reducing agents such as pyruvate cycle, and these electrons are then passed on to the molybdenum iron protein by a known mechanism via ferredoxins to the nitrogenase, where each electron transfer is accompanied by the hydrolysis of two ATP molecules which are also bound to the iron protein.